Good afternoon everyone. I am Rajaprabu, Assistant Professor, Department of Neuroscience Technology, School of Allied Health Science, VMRF-DU. Today we are going to see about neuromuscular junction. At the end of the session, you will be able to understand about what is neuromuscular junction, how it's work, and what are the connection between the nerve and muscle, and how it communicate. These things you will be able to understand, and how its work you will definitely able to understand. The anatomical and physiological functions and their works you will be able to understand in neuromuscular junction. If a general muscle contraction, it definitely by a impulse given by the nerve. It origin from the brain, or it otherwise we will say about like it started from the sensory stimulus, then it goes to brain and it process the information and it come back through descending tract, or we say as at motor tract, and it finally reach the point at muscles by the end nerves. It transmit the information or impulse giving the sodium, potassium and some other action potential, everything will take place and it works, and finally the muscle will contract. One neuron will transmit another neuron or a muscle or any gland by signaling the or transporting through chemical substance called neurotransmitter. A neurotransmitter may be acetylcholine or a dopamine or norepinephrine or etc. It may be anything which will stimulate the neuron or a muscle or a gland, anything it will be. The site of the chemicals interplay in the place of synapses. We called it synaptic cleft, and where it starts or end, we call it as presynaptic knob, and it travel through synaptic cleft and it reach postsynaptic cleft. In presynaptic, it releases the vesicles which contain neurotransmitter or a chemical substances which will induces the muscle or a gland, etc. In synaptic cleft, it travels and reaches the postsynaptic cleft and stimulate the membrane. It's opened up the channels, the acetylcholine or norepinephrine, which will go and attach to the channels and open, and it activate the action potential in the muscle or gland. So it will do the further process, like the muscle will do contraction, or a gland it will secrete a hormone or anything. If a cardiac muscle, it will contract the cardiac heart, etc. So first a sensory impulse travel through somatic motor neuron. It goes through first order neuron and it ascends as a sensory tract and reach thalamus, and then go to somatosensory area in the brain, and it come back through process and come back through descending tract or motor tract. It reaches spinal cord and it will give an impulse to muscles, and the muscle contraction will be take place. In the end, you will able to see an axon terminals. So this is an axon from nerves. You will able to approach the descending tract. Presynaptic cleft will be attached to the muscle, that is the end terminals of the neuron. It will connect to muscles. The muscles, they have postsynaptic cleft which will have a membrane, and the membrane, they are a bilipid layer. They have channels. The channels will be open up through acetylcholine. It will bind to the acetylcholine channel and it open, and it transmit the sodium, potassium in ECF and ICF, and it start generate the action potential. The physiological and anatomical of neuromuscular junction, it end in motor end plate and synaptic motor. This is a motor end plate. This is an axon of a nerve, and this is the terminal button. We call it as terminal button. This is our vesicles which contain a neurotransmitter. It will be the terminal button. It contain vesicles which carry a neurotransmitter, a chemical substance, which will induces this channels, sodium potassium channels. This is synaptic cleft, this is presynaptic cleft, this is postsynaptic cleft. This we call it as subneuron cleft. Subneuron cleft we call it is the red color dotted, we call it as acetylcholine. Acetylcholine is a combined structures of acetyl coenzyme A and choline. It combined and it produce a chemical reaction happen and it produce acetylcholine. This is an axon and this is an axon terminal. The nerve impulse will be come through this, and this will be a myelin sheath, This will be nodes are drawn where the action potential come like a travel of a sodium and potential in between a ECF and ICF, and it reach the end terminals of the axon. We call it as terminal knob of neuron, and it stimulate the calcium ions, and it's entered through the channels and bind to a vesicles. A vesicle also made up of a bilipid layer. It go and attach to the presynaptic cleft and open, and it releases the neurotransmitter. Here we seeing is acetylcholine. So acetylcholine is formed by acetyl coenzyme A and choline. So they form combined and produce acetylcholine. So it will stimulate the postsynaptic membrane channels, and it induce sodium ions to enter. So it will start at the muscle contraction. So acetylcholine will be in the synaptic cleft. The acetylcholine will be in the synaptic cleft. It bind to channels which present in postsynaptic membrane. So what it will happen will be, after the impulse travel through the membrane, the acetylcholine will be come to synaptic cleft, and it will reabsorb by the presynaptic membrane through acetylcholine esterase. It will be esterase, and it separate into acetate and again choline, and it will reuptake taken by the vesicles, and it again the old process will be started. Acetyl coenzyme A and choline combined and form acetylcholine. So the process will be further goes on. So this will be a motor end plate, and this will be sodium potassium channels. This one is presynaptic membrane, this is postsynaptic and synaptic cleft. So everything we will able to see. Motor end plate, this one is motor end plate, this is synaptic gutter or synaptic cleft. So this is a synaptic cleft, postsynaptic terminal, presynaptic terminal, this one is presynaptic terminal, postsynaptic terminal, this one is postsynaptic terminal. Synaptic cleft or synaptic space, acetylcholine will travel from a pre to post, a space will be called synaptic cleft, and subneuron cleft, this here is subneuronal cleft, and acetylcholine in the vesicles, and a red dotted which are called acetylcholine esterase, which will be secreted from here, and it esterases the acetylcholine and reuptake into acetate and choline. In axon terminal, it carries around 3 lakhs vesicles which contain neurotransmitter acetylcholine. So it will be the main chemical substance which will stimulate the another muscle or a gland, etc. we will see in the beginning. In synaptic cleft, it will be the size of 20 to 30 nanometer space in between axon terminal and the muscle membrane. It contains enzyme cholinesterase which will destroy or split the acetylcholine into acetate and choline. Synaptic gutter or synaptic trough. In muscle cell membrane which is in contact with the nerve terminal, it has many fold. We call it as subneuronal cleft. It will be presented in postsynaptic folded like structures. You able to see in here. Here you able to see the cleft. We call it as cleft. Here only it present. We basically call it as subneuronal cleft. So the folded, why it folded mean it needed many surface area. So if it's a flat surface, we unable to place the channels very low. If it will be folded, we able to keep many channels in that. So naturally it will be in a folded form. We called it as subneuronal cleft. The neuromuscular junction consists of what we seen as a postsynaptic cleft and presynaptic cleft and synaptic cleft. The ACH is synthesized locally in the cytoplasm of nerve terminal. We see a presynaptic cleft or a terminal knob. In that it will be produced by acetyl coenzyme A and choline. This is rapidly absorbed into synaptic vesicles by acetylcholine esterase. The synaptic vesicles themselves are made by the Golgi apparatus in the nerve soma cell. Soma means a cell body of the nerve. Here they mention in nerve. Then they are carried by axoplasmic transport. There will be a mini motor transport protein will be there. This is a vesicles. This is axon. Here we is able to approach soma or a cell body. In Golgi apparatus, the protein will be combined and process will be started, and it transmitted through axoplasma through motor proteins, and it take over to the end terminal knob, and the terminal knob they are in the vesicles which are made up of bilipid layer. The axoplasmic transport to the nerve terminal which contain around 3 lakhs vesicles. ACH is synthesized locally in the cytoplasm of the nerve terminal from the active acetate acetyl coenzyme A and choline. Then it combinedly called acetylcholine. Then it will in the vesicle it will releases to the synaptic cleft, and it go and bind to the postsynaptic, and it travel through synaptic cleft, and then it reabsorbed it after the action potential stimulated in the muscle or a gland. It will actively reuptake by acetylcholine esterase. The synaptic vesicles themselves are made by Golgi apparatus in nerve soma cell body. Okay. When a nerve impulse reaches the nerve terminal, it opens calcium channels. So it go and bind to the vesicles, and it diffuses from ECF mini extracellular fluid, and it go to the axon terminal. The Ca++ release the ACH from vesicle by a process of exocytosis. Yeah, the releasing the vesicle will be in axon, and it will come out through exocytosis. Here you will be able to see some small pouches like structures, and they are in the vesicles, and it will come out through exocytosis. So from here to here it will come through, the process is called exocytosis. One nerve impulse can release up to 125 acetylcholine vesicles. It normally contains 3 lakhs. A single impulse will stimulate 125 ACH vesicles. Simultaneously, a impulse will be reaches the postsynaptic cleft. The quantity of release will be one nerve impulse more than enough to produce a single vesicle will be able to initiate the another action potential in the muscle or gland, and it will act in end plate potential, or we call it as end plate potential. AC is combined with the receptor to the subneural cleft. It is open sodium channel. In that channel that will transmit sodium and potassium. The sodium will be going inside and potassium will come out. 4 sodium will go in, 3 potassium will be come out. Sodium diffuses into the muscle and causing a potential which we call it as end plate potential or EPP. An EPP trigger muscle action potential, and in the muscle they are actin, myosin and microfilaments will be there, trypsin, tryptokinin. They are able to make a muscle contraction. After ACH act on the receptor, it will be hydrolyzed by the enzyme we see in before acetylcholine esterase or choline esterase. It will separate the acetylcholine into acetate and choline. The choline is actively reabsorbed by the nerve terminal used for again the production of ACH. The whole process is taken 5 to 10 milliseconds. The overall process will be happened in between the 5 to 10 milliseconds from the process of presynaptic vesicle and synaptic cleft and postsynaptic membrane. So the from pre to post it will take 5 to 10 milliseconds. Even it will reuptake by the choline esterase. Until now we able to see what is a neuromuscular junction and how we transmit from post to pre to post, and how we travel, and what is the function we see. The main function is to contract the muscle, skeletal muscles or a cardiac muscle, anything. So it have ACH receptor in postsynaptic. So it will induces the next stimulation, and in pre there will be a vesicles. So in vesicles only release ACH, so it will go and bind to post. ACH like or mimicking drugs will be there to stimulate the ACH receptor. Some of the drugs will be methacholine, carbachol and nicotine. Actually when you take a nicotinic gum or any other substance for a nicotinic product like tobacco or any other, it will induces the postsynaptic and it will stimulate or it make us more active. So they are mimicking the acetylcholine and it bind to the postsynaptic cleft and it stimulated the postsynaptic membrane. Act for minutes or hour and not distracted by the choline esterase, because they are mimicking substance, they are not acetylcholine. Drug that stimulate the NMJ by inactivating acetylcholine esterase. So they are continuously stimulating. So if acetylcholine esterase will be there, the acetylcholine will be produced through vesicles, but the acetylcholine esterase will be destroyed. So there will be a lack of acetylcholine to stimulate the postsynaptic membrane. So if you are giving neostigmine or physostigmine, they are used in myasthenia gravis and Lambertitan. There will be two diseases called it as neuromuscular diseases. They are occurred mainly due to neuromuscular disorders. So in pre and post, acetylcholinesterase will be reduced the ACH, so it failed to induce the postsynaptic membrane. So if you give in neostigmine or physostigmine, it will inactivate the acetylcholinesterase. So it reduced the destruction of acetylcholine. So it rapidly induces the or initiate the next potential in post membrane. So for example, neostigmine, physostigmine, diisopropyl fluorophosphate. We call it as nerve gas poison. It inactivates acetylcholinesterase for a week. So can cause a death because of respiratory muscle spasm. So it will rapidly constrict, if it act in a respiratory muscles, it rapidly constrict the bronchial cells. So it will constrict due to the constriction in the respiratory cells. It unable to breathe, it make a spasm and lead to death. Drug that block transmission of neuromuscular junctions, curare and curariform like drugs. It will go and bind to ACH receptor. So it reduced the action potential or impulse in the postsynaptic cleft. So what happen will be, it act by competitive inhibition to ACH at its receptor and cannot cause depolarization. So in action potential there will be a repolarization, depolarization, repolarization, hyperpolarization. So it will be like first in -40, in -70 resting membrane potential, it will go depolarization and repolarization and hyperpolarization. So action potential will be in a resting membrane potential, depolarization, it reaches the action potential and it will repolarization, hyperpolarization and again resting membrane potential. So depolarization, repolarization, hyperpolarization. So here diffuse of sodium, potassium, this will happen. And before we see, here you are able to see acetylcholine enzyme A, and this will be taken in a neuron will be this will be nucleus, and this will be a myelin sheath, this will be axon, and this will be end plate terminal knob, and here will be muscle, and this will be, we call it as neuromuscular junction. In that it will be like presynaptic, postsynaptic and synaptic cleft. Here the vesicles will be present. The vesicles, it contain neurotransmitter acetylcholine. The acetylcholine will be formed in the nucleus and axoplasm, and this will be acetylcholine A plus choline. Here what you will be able to see, acetylcholine enzyme A and choline. In after reaches the synaptic cleft and induce the postsynaptic, here already acetylcholine esterase will be there, and it will act in the ACH acetylcholine, and it destroy into acetylcholine, it destroy the acetylcholine into acetate and choline. So these are the things today we are able to see about the neuromuscular junction and what are the drugs which will be a mimic or a stop the neuromuscular junctions process. Thank you for watching my class. Thank you.